Hello my dear students, I'm Dr. Vaishali Bharambe. I've been teaching anatomy for more than 25 years and I love it. Today I'm going to talk about the cell. You should know that I have taken cell part 1 earlier, which is also released on YouTube and elsewhere, in which we've discussed nucleus and plasma membrane. Today we're going to talk about the cell organelles. Okay, so take a look at this image now. You can see here is the cytosol. What is cytosol? Cytosol is the cell fluid, the matrix in which all these organelles exist. So, peroxisome, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi Apparatus, all these are organelles about which you're going to learn today. So, let's begin by talking about mitochondria. Yes, mitochondria is something you've been taught in your school years, isn't it? The powerhouse of the cell. Yes, so let's learn about the structure. Mitochondria is a cigar-shaped organelle having an outer membrane, an inner membrane and an intermembranous space. The inner membrane, as you can see, is folded into a number of cristae and inside which there is an intercrystal space containing a matrix. The matrix contains enzymes as well as DNA, RNA and also contain mitochondrial granules. Now, why is there some data on the screen? The data will be there on the screen so that you can pause and sometimes read the notes. I'm definitely not reading them when I'm teaching you, but it's there for you to, follow as notes. Right. So what does the mitochondria do in your cell? Of course, we know that it produces the power. Okay, so it is the powerhouse of a cell. It makes ATP. It uses glucose to produce energy in the form of ATP. What is ATP? ATP is the energy currency of the cell. I want you to note this. It's the energy currency of the cell. It helps in breaking down of fat. It is also somewhere involved in the programmed cell death. What is programmed cell death? When the cell needs to die so that newer generational cells can come in. Okay, mitochondria is involved in that as well. And it is involved in regulating calcium levels of certain cells. So these are all the functions of mitochondria. There's something very interesting. I want you to think about it. Just pause and understand that mitochondria have their own DNA. So you must be thinking, so how does it matter? Because it has its own DNA, it is capable of replication, self-replication. Okay? How can that be? Isn't it part of the cell? Should it not multiply as per the orders of the nucleus? No. Why so? Because mitochondria is considered to be probably a bacteria that made its way inside our cell. Okay? And it decided to live in the cell. So what did it give the cell in return? It gave the cell energy in return. So in each of your cells, there is a little bacteria sitting in a symbiotic relationship with the cell, getting shelter from the cell, giving energy in return. Isn't that interesting? I find it enchanting. Right. Now I want you to think about mitochondrial cytopathy disorders, which basically means that there are some mutations in the mitochondrial DNA. Mitochondria, energy. Mutation in DNA, lack of energy. Very simple. Okay, broadly speaking, that is what you should be thinking. So, Kearns-Sayre syndrome, MELAS, all these are examples of mitochondrial cytopathy syndromes. Okay. This is a note for you. You can pause the video and understand that in each of these syndromes, MELAS, Lee's syndrome, Kearns-Sayre syndrome, in Pearson, what are the symptoms you would see? It's not under the purview of mitochondria topic to discuss this. However, please feel free to pause the video and read this. There's something very interesting called mitochondrial inheritance. Okay. Now, the boys we want. Mitochondria in any individual is always obtained from the mother. See, there is an oocyte and there is a sperm, right? The sperm, as it enters into the oocyte, gives the oocyte only the nuclear content. Whereas the oocyte itself has its own cell contents as well as its own nucleus. So from the father, you have got nuclear content. From the mother, you've got cell as well as nuclear content. So the mitochondria that you have or anybody has is always maternal in nature. The mitochondria is always maternal in nature. So now imagine that the mitochondria is having some disease. It will pass from the mother, take a look. It will pass from the mother to the daughter and son. It will pass from the daughter also to her daughter and son. But can it pass from the son? It cannot pass from the son. Why? Because the son is giving only nuclear content. Son is not giving mitochondria to his progeny. This is called as mitochondrial inheritance, where all inheritance happens through the female, through the mother. Okay? Is that interesting? I hope you loved it as much as I do. Okay. So we now go to the next cell organelle called as ribosome. Ribosome, think protein production. The moment you come to the word ribosome, think protein production. These are tiny structures that make proteins. They are miniature factories which, according to the RNA instructions, assemble amino acids together to produce proteins. Okay? So, DNA produces mRNA in the nucleus. See how it goes, okay? In the nucleus, the DNA produces mRNA. mRNA comes out, ribosome reaches the mRNA. Ribosome reads from it and begins to produce proteins using amino acids. This is the way in which amino acids are used to produce proteins. Right. Ribosomes can be found floating freely in a cell. They can be found attached to the endoplasmic reticulum, in which case it's called as rough endoplasmic reticulum. We'll learn more about it soon. So they are crucial for producing proteins which are required for every activity of the cell. Without ribosomes, the cells would not be able to produce protein and would not be able to maintain your growth, repair as well as maintain the cell itself. So that's ribosomes. Remember, this is a very brief introduction. There is a lot more you should be reading, but this is just to give you a very quick idea about each of the cell organelles. Okay, now stop. Take a deep breath, take some water or drink some coffee, relax your mind. Don't allow matter build up in your brain. It gets stressful and you stop enjoying the whole subject. Medicine is to be studied only by enjoyment, only by fun, only by wonderment. I hope I can transfer to you the wonderment of studying medicine. That's the aim of these lectures. I hope you enjoy them. Alright? So I'm also taking a pause. I'm also drinking coffee. Come on, go get your coffee. We'll go on with the rest of the lecture. Right. So let's continue. Okay. So here you're seeing something called as an RER, rough endoplasmic reticulum. Okay. On it you can see tiny fixed ribosomes. Can you see those dots? Okay. And then here you're seeing some free ribosomes. So that's how ribosomes are. Free ribosomes attached to RER ribosomes. Okay. Right. So let's come to something called as an endoplasmic reticulum. Okay. So it can be in the form of tubules, it can be cisternae, sacs, etc. A little bit of history. It used to look like some kind of lace-like network in a cell. Remember, we're talking about long time ago when we did not have fancy microscopes. So it used to just look like some kind of network. So it was called endoplasmic because it was in the cell. It was called reticulum because it was looking like some kind of a mesh. Okay. Now we know much more about it. It was found that this mesh or this network could be smooth endoplasmic reticulum, could be rough because of ribosomes studied on it, rough endoplasmic reticulum. It's the same thing. Okay. Now, all of them were continuous with each other and were continuous with the nuclear envelope. Okay. So here we are looking at rough endoplasmic reticulum. Here we are looking at smooth endoplasmic reticulum. Little bit about it. So it's a system of, use this beautiful cell here to understand. Can you see how much rough endoplasmic reticulum there is? I want you to note all these, okay? So the outer surface is rough because of ribosomes. Inner surface, as you can see, is quite smooth. That's the space, okay? Rough endoplasmic reticulum produces proteins which are released in its own cavity. Later on, they will be removed from this, they will leave rough endoplasmic reticulum in the form of tiny vesicles called transport vesicles. We'll learn about them. So, in those cells where you want to produce a lot of protein, you will have a lot of RER. Come back to the screen now. Can you see this is labeled as a pancreatic assigner cell? Pancreatic assigner cell. It's going to produce enzymes which are used for your digestion. Enzymes, proteins. Therefore, this cell has to have a lot of RER. Can you see how much RER it has? Okay. Of course, for production, there is energy requirement. So, you can see a lot of mitochondria sitting close by to provide energy. Isn't that exciting? So much happening inside your pancreatic cell. Right. So, that's all the RER. You can see here, these are all the pancreatic cells, and you can see how much RER the cell is shown to be having. Right. Just a little note. Remember, any cell that has a lot of ribosomes and lot of rough endoplasmic reticulum is able to absorb a lot of hematoxylin. A cell, any slide is usually stained with a H&E staining, hematoxylin and eosin staining. So if it contains a lot of protein containing organelles, it stains bluish in color. So when you're looking at a slide, if it looks bluish, think lot of protein produced. Okay, think that. This note is for that. You can read it at your leisure. Again, I keep some notes on the screen. I'm not going to read them. You can pause and study. Right. So cells where you will find a lot of RER which look hematoxylin or basophilic will be plasma cells, pancreatic assigner cells. Okay. Now here you are looking at a hepatocyte. Look at it. What is special about it? It's a hepatocyte, it's a liver cell. Liver is the seat of metabolism in your body. So much metabolism happening in your liver. See how much RER, see how much SCR, okay? See how much mitochondria. So this one cell should give you an idea about how these organelles function in cells, okay? Right. So smooth endoplasmic reticulum is another network of tubules, but it helps in synthesis of lipid and steroids. So wherever you want to synthesize lipids, you want to synthesize steroids, again liver, you will find a lot of smooth endoplasmic reticulum. Rough endoplasmic reticulum produces proteins. It is transferred to the smooth endoplasmic reticulum, which synthesizes lipids on it. So lipoprotein complex is produced. This lipoprotein complex is then delivered to the Golgi apparatus. Yes, we are going to learn about it next. The Golgi apparatus will then package it, okay, and this packaged lipoprotein complex, okay, will be released from your cell. Think about it. Okay, so like I said, we go to Golgi complex. Protein was being produced by ribosomes, by rough endoplasmic reticulum. Lipids were being produced by smooth endoplasmic reticulum. What is Golgi complex doing? Golgi complex is the packaging industry of your cell. Okay, so whatever you produce, it has to be nicely packaged. Okay, who is doing the packaging? Golgi complex. Welcome to the packaging factory. Come. So now here is your Golgi complex in the same cell. My suggestion to you right in the beginning of my lecture 1 was draw the cell. I suggest you still draw the cell. I have drawn this, it's not difficult at all. Try it out. Otherwise, take a simpler diagram, but draw it, okay? Right, so that's the Golgi complex. Now, I have drawn these rather simple line diagrams. You must be thinking why they're so simple. They are simple because I want you to draw them. That's why I can give you a fancy diagram, but then you won't find them easy enough to draw. So please draw this. Right, so Golgi complex initially was considered to be a staining artifact. something is there in the cell, because microscopes were not good, remember? Later on it was found that it's actually a cell organelle. It's made up of 2 to 4 stacks of, sac kind of structures, one on top of the other, as you can see over here. Something like saucers kept one on top of the other. Each Golgi complex has a cis phase. It has a medial Golgi. It has a trans phase. All sounding Greek and Latin. Yes. So we drink coffee. Right. So what is this cis phase? Okay, take a look. So this is your rough endoplasmic reticulum. It is releasing produced protein in the form of these small vesicles called transport vesicles. The cis Golgi is receiving them. Okay. So cis is the phase that receives material from rough endoplasmic reticulum, passing it through the medial Golgi over here. This material gets packaged and reaches the trans Golgi. Trans Golgi is that phase that is facing the cell surface. See, this is the cell surface. So this material is packaged and reaches the trans Golgi. At the trans Golgi, it is released. Okay, you can see it being released over here onto the cell surface. So cis Golgi, medial Golgi, trans Golgi. 3 parts of Golgi complex. How is protein produced? RER will produce protein? Yes. Release it in the form of transport vesicles. Reach cis Golgi. Transport it to the next, maybe medial Golgi. Reaching the trans Golgi. Released from the trans Golgi after addition of oligosaccharides to it, packaging. Okay? And glycoproteins are formed. These will be released in the form of secretory vacuoles onto the surface. Okay, let's look at this beautiful image to understand. So here's your transport vesicle, here's your cis Golgi receiving it, here's your trans Golgi releasing it after packaging, covering it with oligosaccharides. This is your secretory vesicle. So secretory vesicle knows I have to travel to the cell surface, I have to fuse with the cell surface. See what's happening here. I have to fuse with the cell surface and I have to release my content out of the cell. Okay. So by which process is it released? Yes, it's released by exocytosis. Right. So this is how the whole cell functions. Okay. And various organelles are involved, interconnected in their activities. So what does Golgi complex finally do? We know it's a packaging industry, adding carbohydrates to produce glycoproteins, adding oligosaccharides to lipids to produce glycolipids. See, it is membrane structure, membranous structure. So, it is involved in membranous synthesis and recycling also. Fantastic, it produces primary lysosomes. That's the next topic we're going to discuss. Very exciting. I hope you enjoy it. So, we've covered a lot of organelles. I'm not going to read all this. But the next thing we're going to discuss is lysosomes and peroxisomes. Lysosomes are highly dynamic. Pay attention. Again, have a sip of coffee, maybe some water. Relax into the class. Okay. So, we've heard about a lot of membrane-bound structures or vesicles. Like I told you, there are storage vesicles, there are pinocytic. In lecture 1, I mentioned pinocytic vesicles, phagocytic vesicles, exocytic vesicles, transport vesicles. Similarly, there are storage vesicles, lysosomes and peroxisomes. What does it store? Come take a look. So, here is your peroxisome, here is your lysosome. Lysosome contain enzymes. Lysosome contain enzymes. These enzymes can be used to destroy unwanted material in the cell. Heterophagy. This material could be ingested from outside. I could have gone outside, brought something in my cell, and now it's in my cell and I don't want it. Okay? When that is destroyed, it's called heterophagy. Whereas autophagy, when some cell organelle has been working for so long, it's tired, it has to be destroyed. Okay, it has reached the end of its functioning capacity. When you destroy the cell material itself, it's called autophagy. See, autophagy is a term which is repeatedly being used today. So many people say you have X kind of diet, it increases your autophagy. I said X, I did not say which diet. That is for you to find out. Okay, so how are lysosomes formed? Rough endoplasmic reticulum produces enzymes. These are released by transfer vesicles. Okay, Golgi Complex covers it up. Okay, and a primary lysosome is formed. If it contains enzymes, why don't the enzymes digest the lysosome itself? If they are so powerful, should it not digest it? So understand that they are inactive in the absence of acid medium. All the enzymes that are there inside a lysosome can only become active in the presence of particular acid medium. That's why they are there. They are waiting for that moment to occur when they can become transformed. Okay? So, primary lysosome at one moment fuses with an endosome. Okay? It can be pinocytic vesicle, it can be phagocytic vesicle, anything. Fuses with it, and now a secondary lysosome is formed. This secondary lysosome has got activated enzymes which can digest fluid from the pinocytic vesicle, solid material from a phagosome, or it can digest material in an autophagic vacuole. Basically, the lysosomal enzymes activated, they will go on fusing with different types of vesicles in the cell and destroying whatever material is there in them. They are directed to go and fuse with only that or vesicle which needs to be destroyed. So, material gets degraded inside the lysosome and it can remain in the cell in the form of what is called residual body. Or it can remain in the cell as cell inclusions. See, as we age, our brain cells have an increase in a particular granule called lipofuscin granule. Okay, cells begin to look yellow. So that's the lysosomal enzyme which is digesting material which is getting collected in your brain. Or in fact, neuromelanin that you see in substantia nigra, again in the brain, that's also an end product of these enzymes. So the material otherwise can also be removed from the cell by process of exocytosis. Question: can lysosomal enzymes leak out? Yes, interesting question, isn't it? Yes, they do leak out. Normally, they don't want to leak out. They are inside vesicles. But if you're going to expose your body to ionizing radiations, to carcinogens, okay, or to anoxia, this will lead to breakage of lysosomal enzymes will be released outside, leading to cell death. So be warned, these are the effects of ionizing radiations. Right. So what is lysosomal storage disease? See, lysosome is containing enzymes. Suppose there is something in those bunch of enzymes that they are not able to destroy. Okay. Suppose there is a defect in the enzyme. Then the byproduct of the breakup is unable to get out of the cell and begins to build up inside the cell. Okay. Then that is lysosomal storage. Okay. So that is called as a lysosomal storage disorder. So basically there's a deficiency or malfunctioning of lysosomal enzyme, and the substrate of that metabolism begins to, or waste material begins to collect in the lysosome. Okay. Again, I'm giving you a list of diseases which are typically lysosomal storage disorders: Gaucher's disease, Tay-Sachs disease. Remember, often these are inherited, genetic disorders. You don't suddenly acquire lack of an enzyme, So usually it is some gene is carrying it and it is manifested in the child. This last bit about peroxisomes. Peroxisomes are vesicles that contain oxidative enzymes important for oxidative detoxification within the cell. So now we've learnt about a lot of cell organelles today. Then what is left? Ma'am, we're done with nucleus, plasma membrane, we're done with the cell organelles, or we're done with the cell. No, we're not done with the cell. Remember, the cell is having a cell skeleton. Like you have a skeleton on which your muscles are built, the cell has a skeleton on which the cell is built. That is why the cell has a shape. So, cell skeleton, who produces the microtubules, microfilaments which create the skeleton? What are cellular projections like cilia and microvilli? What are centrioles? Okay, that's what we will be discussing in our next lecture, Cell Part 3. Do watch Cell Part 1, do watch Cell Part 3 to understand the cell as a whole. I hope you liked this lecture. If you liked it, do like and subscribe. I would love it if you gave me some suggestions about how more my lectures can help you study and enjoy your journey in medicine. Okay, so I'll see you in my next lecture. I hope you enjoyed this one. Bye.